Hello and welcome. Let's go through chapter 5 today. So today when we go through chapter 5 takes us into cell division. So here we're going to look at some fascinating stuff in relation to the cell. Now when we talk about the cell, you can, if you haven't already seen all the other fascinating stuff that we've been talking about, we're adding more to it there. So when we go through now and talk about the cell, today now we're going to start with the cell cycle. Now, we're going to see how the cell can go and divide into two different cells. And then we'll see that will be one type of a division we're going to go through and we're going to look at. And then there's going to be another one where we're going to see one cell is going to go through one division, become two cells, and the two cells will then go through divisions themselves and then give rise to four different cells. So we'll look at basically both of these different processes. So first here, I want you guys to know when we start life as a single-celled fertilized egg. We start life as a single-celled fertilized egg, and then by the process of cell division, we grow into organisms that contain trillions of cells. Cells are going to divide constantly throughout life to make new cells. Then you have this term apoptosis, okay, which we'll talk about in a few minutes then. Now, when we go through and we talk about the cell cycle, I'd like you to know that the cell cycle is going to be a series of steps. It's a series of steps that's going to take place in order for a cell to divide. It's a series of steps that are going to take place in order for cells to divide. Now, when we talk about somatic cells, we'll see they're going to be the body cells that are going to continue to undergo cell division even as an adult. And we'll see thousands of new blood cells, skin cells, cells that line the digestive tract, respiratory tract, are going to be produced every day. They're going to be produced every day. Now, when we talk apoptosis is going to be programmed cell death. Now, this process, apoptosis, what it does is it decreases the number of cells. This process of apoptosis is going to decrease the number of cells. It occurs during growth and development. And it occurs during growth and development to remove unwanted tissue. It also plays an important role in preventing cancer. Apoptosis also plays an important role in preventing cancer. Now, when we talk about apoptosis occurring during development to remove unwanted tissue, we can see that happening in the embryo. The embryo has webbed hands and feet, and apoptosis is going to go and remove them from one another. So we said it also plays an important role in preventing cancer. An abnormal cell that could become cancerous will die via apoptosis, preventing cancer. The cell cycle is a series of steps that takes place in order for basically cells to divide, as we can see. Now the stages we're going to see are going to include interphase and then the mitotic stage. Now when we look at the interphase, when we look at interphase we can see is divided into three stages. It's divided into three stages, and we'll look at each of these three stages in greater details in a few seconds. We'll look at the three stages in greater detail here in a few seconds. And then the mitotic stage we can see is also subdivided. So let's talk about first the stages of interphase. Now when we talk about interphase, you can see most of the cell cycle is going to be spent in interphase. Most of the cell cycle is going to be spent in interphase. So here we can see that. Here's the cell cycle. So here we've got interphase first, we said, right? And then we said we have the mitotic stage. So interphase and mitotic stage. And then we said each of them are going to be further subdivided. So here you can see, this is the cell cycle. So here from this part right in here where G1 begins, right, you can see all the way around, this is all interphase. And we said interphase, here you can see it ends at G2. And interphase we said is going to be subdivided. So here you can see the subdivisions there. And then here we've got the mitotic stage. And the mitotic stage is going to consist of mitosis, we said, and cytokinesis. Well, you'll see that right there. So I'd like you to know, you can see most of the cell cycle is spent in interphase. Now during this phase, the cell is getting ready to divide. During interphase, the cell is getting ready to divide. It grows larger, and the number of organelles and DNA doubles. And the number of organelles and DNA doubles. This phase, as we can see, is divided into 3 stages. It's divided into 3 stages. The first stage is G1 stage. G1 stage, we can see here, is going to be the stage which is going to occur before DNA replication. The G1 stage is a stage that occurs before DNA synthesis. Then you have the S stage, okay, and we'll look at that then in greater detail. So here you can see, now in G1, I'd like you to know the cell doubles its organelles, we said, and it accumulates material for DNA synthesis. And it's going to accumulate material for DNA synthesis. So it's getting ready for DNA synthesis. Now I'd like to know there's also G0. Now here we can see in G1, we can also move to G0. So from here we can go to what's called a G0. And then the cell can come back from G0 into G1. And don't worry about this G1 here we see. We'll come back and we'll talk about this checkpoint in a few minutes. So here, G1, you can see again, we said the cell doubles its organelles, right? Organelles we've gone through, we've talked about organelles, so you know what I'm talking about when I'm mentioning organelles. And it accumulates material for DNA synthesis. So G0 then is going to occur in some cells. It occurs in some cells, such as nerve and muscle cells, which do not complete the cell cycle. So here, G0 is basically a stage where the cell becomes kind of arrested. Nothing is going to happen there, okay? Now when we move to the S stage is going to be basically, you'll see a stage we said where DNA synthesis is going to occur. So it's going to include DNA synthesis. So here we'll see, where do we go? Each chromosome consists of 1 DNA molecule. It's called a chromatid. Now after DNA replication, so after this S stage, okay, so once we're here at the end of this S stage, before we begin the G2 stage, okay, here you're going to see after DNA replication, each chromosome consists of 2 sister chromatids. 2 sister chromatids. Now the term sister, you're going to see, we're going to use a lot. It's nothing, it's just a term that they use. We're going to also use the term daughter cell a lot as well. And you'll see that's going to be the cell that's going to be formed after, as we saw here, one cell, okay? The cell cycle is an orderly sequence of events that's going to occur between the time a cell divides and the time the resulting daughter cells divide. So the daughter cells are going to be the cells that are formed when the cell divides, okay? So it's not just a female or anything like that. It's just the reference term that they use. And then we can move to the G2 stage. When we talk the G2 stage, then here we'll see G2 stage is going to occur after DNA synthesis. It'll take place after the S stage, right? Now here when we go through and we talk G2, the cell synthesizes proteins for cell division. It's going to start synthesizing proteins for cell division. And synthesizing proteins, we're going to see, is going to be it's going to be a very important event that takes place, and we'll check it out as we progress. Now, when you think of G for G1, G2, you can think of the G there as a growth stage. You can think of it as kind of a growth stage. During G1, the cell doubles its organelles. Some other cells, we said, enter G0 and become arrested. Okay, they don't complete the cell cycle. And then some cells can come out of the G0 phase and complete the cell cycle, and some don't even enter it. So you have variations that take place there. Now, when we talk then S stage, you saw DNA replication take place, and then G2 stage, the cell synthesizes proteins needed for cell division, we said. Next then here, let's talk about the M stage. Now we talk M stage. So here we can see then the M stage, or the mitotic stage, is going to include mitosis and cytokinesis. So when we go through, we talk mitosis, you're going to see mitosis is going to be a division of the nucleus now. A division of the nucleus. So think about that. Now we're dividing the nucleus, right? What if we started dividing the nucleus before? That would be kind of a little bit difficult because we have barely synthesized the DNA that the new cell is going to need in order for it to function, right? DNA is where all the orders for the proteins and all the various components in relation to the human is going to come from, right? That's where all the kind of instructions for protein synthesis are going to come from. So without having this DNA, the new cell is going to be then useless. So this nice orderly sequence of events, as we described, is almost perfect. So here when we talk then the M stage, mitosis and cytokinesis. Now when we go through after interphase then, so after interphase, the cell enters the mitotic stage. It enters the mitotic stage. Now this stage not only includes mitosis, which is going to be, as I described, the division of the nucleus and the genetic material, but also it includes, you can see, cytokinesis. It includes cytokinesis, which is the division of the cytoplasm then. Which is the division of the cytoplasm. During mitosis, the chromosomes separate and distribute into two daughter or new cells. The chromosomes separate and are distributed to two daughter cells or to two new cells. Mitosis, okay, we can see there. And then the division of the nucleus, it follows the interphase as we described. The sister chromatids are going to separate. They're going to separate into daughter chromosomes. And then they're going to be distributed to two daughter nuclei. Cytokinesis, the division of the cytoplasm, it follows mitosis, where the two daughter cells that are identical, where then we're going to form two daughter cells that are identical to the mother cell, where we're going to form two daughter cells that are identical to the mother cell. So here then we can see the events that are going to take place in apoptosis. Here we've got a nice view of some normal cells, and then we'll look at one cell and we'll see what basically happens to the one cell. So this one cell, we'll see what's going to happen is the cell's going to start to round up and the nucleus is going to start to collapse. And here you can see chromatin condenses and nucleus starts to fragment. And then here we see the plasma membrane starts to blister. We get these blebs basically that start to form. And then here you can see the cell starts to break apart. And then here we've got the cell fragments. And then you can see they're going to contain DNA. And basically the cell is then useless now. So when we talk apoptosis again, it's a typical series of events that brings about cell destruction. And caspases, these are enzymes. These are enzymes that are going to be responsible for apoptosis taking place. They're held in check by inhibitors. These caspases, they're held in check by inhibitors, and they're unleashed by internal and external signals. They'll be unleashed by external and internal signals. I'd like you to know, when we talk about the control of the cell cycle in cancer, we'll see the cell cycle is controlled by both internal and external signals. It's controlled by both internal and external signals. Eukaryotic cells have a complex system for regulating the cell cycle. The cell cycle is controlled, again, by both types of signals. Now, the events we talked about in the cell cycle, they have to occur in the correct order. They have to occur in the correct order. So if I was to give you a question asking you to put them in order, you would have to be able to put them in order. M stage comes before G2 or the S, or which stage comes before which, and basically see if you can recall them. So here when we go through and we talk, then you can see, so as we can see, the cell cycle is controlled by both types of signals, internal and external signals. When we talk internal signals, the internal signals, what they're going to do is they're going to ensure that the stages follow one another in a normal sequence. They're going to ensure the events occur in the correct order. They also check that each stage is properly completed prior to the next. They also check that each stage is properly completed prior to the next stage. The cell cycle has three main checkpoints. It has three main checkpoints that make sure everything is fine with the DNA. They make sure everything is fine with the DNA and any damage is repaired. If it's not repairable, apoptosis is going to occur. Then apoptosis will occur. When we talk about the external signals, they're going to tell the cells whether or not to divide. Now, the three checkpoints that control the cell cycle are going to be checkpoints found in relation to the G1 stage, G2 stage, and also M stage. So G1 stage, we're going to see then, we've got basically a checkpoint here. We've got a protein here I'd like you to know about. That's protein P53. Protein P53 stops the cell cycle in G1, and it tries to repair the cell. And if it cannot repair the cell, apoptosis will occur. Apoptosis will occur. So if everything is fine here, the cell then becomes a committed cell and it'll continue forward. The next checkpoint then is going to be found in G2. This G2 checkpoint is going to check basically the cell after the S stage. The G2 checkpoint checks the cell after the S stage. This G2 checkpoint is the mitosis checkpoint. Mitosis will occur if DNA has replicated properly. So it'll move forward only if DNA has replicated properly. Otherwise, apoptosis is going to occur if DNA is damaged and it cannot be repaired. Then we have an M stage checkpoint. The M stage checkpoint. Now this M stage checkpoint is going to check the spindle assembly. So it's known as a spindle assembly checkpoint. Mitosis will not continue if the chromosomes are not properly aligned. And if everything is fine, it'll continue then, and cell division will occur. So mammalian cells are going to enter the cell cycle only when stimulated by external factors. Only when stimulated by external factors. Now, growth factors are signals that set into motion the events associated with entering the cell cycle. Growth factors are going to be signals that set into motion the events associated with entering the cell cycle. For example, We can see when for example, we can see when blood platelets release a growth factor, skin fibroblasts finish the cell cycle then. I'd like you to know that there's two types of genes that are going to control progression through the cycle. They are going to be proto-oncogenes, so two types of genes that control the movement of a cell through the cell cycle are going to be proto-oncogenes and tumor suppressor genes. And tumor suppressor genes. Now, when we talk proto-oncogenes encode proteins. Proto-oncogenes are going to encode proteins that promote the cell cycle and prevent apoptosis. And they prevent apoptosis. So again, proto-oncogenes encode proteins that promote the cell cycle and prevent apoptosis. They're likened to a gas pedal of a car. They can mutate to become oncogenes. Oncogenes are proto-oncogenes that have mutated to become cancer-causing genes. Oncogenes are proto-oncogenes that have mutated to become cancer-causing genes. So here we can see then, growth factor comes into play, binds to the receptor. When it binds to the receptor, it activates these ROS proteins. They then activate the signaling pathway. They'll activate the signaling pathway then, which is going to help produce proteins for cell division. So here we can see then, our proto-oncogenes, we can see are going to code for a growth factor. So here we can see our proto-oncogenes are going to code for a growth factor, a receptor protein, or a signaling protein. Proto-oncogenes are going to code for a growth factor, a receptor protein, or a signaling protein in a stimulatory pathway. Now, if a proto-oncogene becomes an oncogene, we said, the end result can be unregulated cell division. Becomes cancer cells, we said. Becomes cancer-causing genes, we said. Next, when we talk our tumor suppressor genes, our tumor suppressor genes are going to be genes that encode proteins that stop the cell cycle. They stop the cell cycle and they promote apoptosis. So codes for signaling proteins in an inhibitory pathway. If a tumor suppressor gene mutates, the end result can be unregulated cell division. So we talk cancer. Cancer is unregulated cell growth. Unregulated cell growth. So tumor suppressor genes are like the brakes then on the car. They're going to inhibit progression through the cell cycle. They encode proteins that stop the cell cycle and promote apoptosis, we said. Mutations then are going to cause those brakes not to work. And then the gene products do not inhibit the cell cycle like they're supposed to. Carcinogenesis. It's the development of cancer. It's a multi-stage process. It involves disruption of normal cell division and behavior. And then mutation initiates cancer. And then mutations initiate cancer in proto-oncogenes or the tumor suppressor genes. Now we move to mitosis. Let's talk maintaining the number of chromosomes here. Here we'll see when we go through, before we get into mitosis, let's review eukaryotic cells contain chromatin. We said that they contain chromatin. Eukaryotic cells contain chromatin, which is basically DNA and proteins, histone proteins. and it's going to be dispersed in a non-dividing cell. The chromatin will condense into a compact form for cell division. And the compact form then we'll see is going to be into a chromosome. Some proteins called histones, they're going to be responsible. Some proteins called histone proteins, they're going to be responsible for packaging DNA. They're going to be responsible for packaging DNA so that way it can fit into such a small place that it's going to be found in the nucleus. So that way it can fit into that nucleus. And we said when the eukaryotic cell is not undergoing division, that chromatin is going to be dispersed. It's going to be dispersed or it's going to be extended throughout the cell. So at the time of division, at the time of cell division, chromatin is going to coil, it's going to loop, and it's going to condense into a highly compacted form, which we refer to as chromosomes. Each species has a characteristic chromosome number. Human cells contain 46 chromosomes. This number is called the diploid number, 2n. It's called the diploid number because it contains 2 of each type of chromosome. It contains a pair of each type of chromosome. One member of each pair originates from the mother, and the other originates from the father. Half the diploid number, called a haploid number, or 1n, is going to contain only half. It contains only one of each kind of chromosome. In humans, only sperm and egg cells are going to contain haploid number of chromosomes. So let's look at this overview of mitosis. Now, mitosis is a nuclear division in which the chromosome number stays constant. Here, a 2n nucleus divides to produce daughter nuclei that are also 2n. Now, before nuclear division takes place, we saw DNA replication occurs. DNA replication occurs duplicating the chromosomes. A duplicated chromosome is going to be composed of 2 sister chromatids that are going to be held together in a region. A duplicated chromosome is going to be composed of 2 sister chromatids that are going to be held together in a region called the centromere. Sister chromatids are genetically identical, right? They're genetically identical. They contain the same DNA sequences because the DNA that we saw there underwent DNA synthesis, DNA replication. It copied. When it copied itself, it produced an exact copy of itself. So again, when we talk the sister chromatids are going to be genetically identical. They contain the same DNA sequence. At the completion of mitosis, each chromosome is going to consist of a single chromatid. So here if we have the one single chromatid we could see, when it undergoes replication, it copies, we have this formed here. So here you can see the sister chromatids. Here you have one of those chromosomes, and here you have the other chromosome, and they're held together at the centromere. Now, when they undergo division, when they divide, you can see here the division is going to happen, and they're going to separate them. Each daughter nucleus at the end of mitosis, at the completion of mitosis, each chromosome is going to consist of a single chromatid, we said. So each daughter nucleus is going to get a complete set of chromosomes. So here you could see this is now a set of chromosomes. Now these chromosomes, once they undergo DNA replication, we can see here they're all going to get copied. So once they get copied, you can see each one has doubled. And then we're going to have division, mitosis, occur. After DNA replication, nuclear division occurs. And then here you can see each daughter cell will get its own diploid number of chromosomes. So one 2N cell is going to give rise to two 2N cells. Mitosis is a type of nuclear division that occurs when tissues grow or when tissue repair occurs. After fertilization, the zygote divides mitotically. After fertilization, the zygote divides mitotically. It divides mitotically, and mitosis continues during development and throughout one's lifespan. Let's look at then mitosis in greater detail. So when we talk mitosis in greater detail, mitosis, I told you, is a nuclear division. Mitosis is a nuclear division that forms two daughter nuclei, which produces two daughter nuclei. And the two daughter nuclei are going to have the same number of chromosomes and the same kind of chromosomes. Right? The daughter nuclei will contain the same number of chromosomes and the same kind of chromosomes. The daughter cell is going to be same as the parental cell. The daughter cell is going to be the same as the parental cell. So look at here, one red long chromosome, one short red chromosome, one short red chromosome, one long blue and one short blue, one long blue and one short blue. Same thing with this cell. Long red, long red, long blue, short red and short blue, short red and short blue. So again, you can see the daughter cell is the same as the parental cell. A spindle brings an orderly distribution of chromosomes to the daughter nuclei. So here you can see, we're going to see these spindles are going to be basically formed. We're going to look at all of this in way greater detail. This is just a nice condensed version. So here we're going to see these spindles are going to actually be formed, and we're going to have lots of good stuff happening inside of here. We'll go through and we're going to check out. So let's look at then mitosis in detail. So we look at mitosis in detail. Mitosis is going to be a continuous process. It's a continuous process that's divided into 4 phases. It is a continuous process that's going to be divided into 4 phases. If we move back, you can see that right inside of here. So this is where we're at. Mitosis is a process that's divided into 4 phases. Prophase, metaphase, anaphase, and telophase. And we're going to see prophase can get divided into early prophase, late prophase, and mid prophase. So we'll go through and we'll check out all of this good stuff here. So let's look at prophase. Now when we talk prophase, here I'd like you to know the nuclear membrane is going to disappear. The centrosomes are going to migrate. And spindle fibers begin to appear. The chromatin is going to condense, and chromosomes become visible. Each is going to be composed of 2 sister chromatids, as we saw. The centromeres are going to attach to the spindle fibers. And chromosomes have no particular orientation. So here in prophase, we can see it gets divided into early prophase, then prophase, and prometaphase. So basically late prophase, they also describe it as by some authors. So here when we go through, the centrosomes begin moving away from each other. They move toward opposite ends of the nucleus. And we said the nucleus is going to begin to break apart. The nuclear envelope is going to begin to break apart. It starts to fragment. Nuclear fragmentation occurs. Spindles are going to form during late prophase. Spindles will form during late prophase, and the chromosomes become attached. So here you can see we've got our basically asters right inside of here. Okay? And here you can see we've got the nucleus. And here we've got our genetic information inside of here. The chromosome condensing and forming chromosomes. Now here the centrosomes have duplicated. So here you can see the centrosomes have duplicated. Chromatin is condensed into chromosomes, and the nuclear envelope is fragmenting. Here then in prophase, nucleolus has disappeared, and duplicated chromosomes are visible. The centrosomes begin moving apart. They begin moving apart, and the spindles are in the process then of forming. Then here in prometaphase, we can see the kinetochore of each chromatid is going to be attached to a kinetochore spindle fiber. Then polar spindle fibers stretch from each spindle pole and overlap. So here you can see that happening in an animal cell. And here you can actually see these events taking place under a microscope. In a human cell. And then plant cells. The other type of eukaryotic cell we're talking about. So then we can move to metaphase. In metaphase, here the chromosomes are going to line up. They will lie on a plane perpendicular to the spindles. Here the chromosomes line up and they'll lie on a plane perpendicular to the spindles, known as the metaphase plate. Here you can see that right inside of here. So here we can see that right inside of here. The chromosomes lying on the metaphase plate. And when you look at it under the microscope, you can see that's usually one of the more easier ones to find, and then you can start kind of looking around and looking for then anaphase, or you could move backwards and look at then basically prophase and kind of start looking at prophase in greater detail and be able to see each of the different cells in the different stages. So then after metaphase, we move to anaphase. Now in anaphase, the sister chromatids separate. They're going to separate, becoming daughter chromosomes. In anaphase, the sister chromatids are going to part. They separate, becoming daughter chromosomes. Becoming daughter chromosomes that move towards opposite poles. They move towards opposite poles. And that's another one that's usually nice and easy to find under the microscope as well. And then here we can see telophase. In telophase then, we can see the spindle disappear. In telophase then, the spindle disappears. And the nuclear envelope is going to reassemble. And the nuclear envelope will reassemble around the chromosomes, preparing us then for cytokinesis, getting us ready for the next stage, cytokinesis. So getting us ready for cytokinesis. So here we could see all the events. You've got early prophase, prophase, late prophase, metaphase, anaphase, and telophase. So actual pictures from the microscope, and then you can see illustrations. So when we talk cytokinesis in animal cells. Cytokinesis or cytoplasmic cleavage, it accompanies mitosis, but they are separate processes. Division of the cytoplasm begins in anaphase and continues in telophase and completes just before the next interphase. Organelles are also duplicated. In cytokinesis, you'll see a cleavage furrow. It forms between the daughter nuclei. And there's a contractile ring that contracts, helping to deepen that furrow. And this process is going to continue until the separation of the two cells is complete. So here we can see cytokinesis. Now, we can go back to this picture right inside of here where we're able to appreciate then it's starting to begin. You can see that now better illustrated right in this view where you can appreciate the cleavage furrow actually formed. Now what happens is this ring will continue and it'll continue to contract, helping to separate then the two daughter cells. All right, so let's move on to meiosis then. Now, when we talk meiosis, I want you to think reducing the chromosome number. When we talked mitosis, we said maintaining the chromosome number. So you think meiosis, reducing the chromosome number. Now, meiosis occurs in any life cycle that involves sexual reproduction. Meiosis reduces the chromosome number in a way that each daughter nuclei receives only one of each kind of chromosome. So it occurs in the life cycle of sexually reproducing organisms. It reduces the chromosome number in half. Provides offspring with a different combination of traits from that of either parent. Meiosis, we'll see, is going to begin. So at the start of meiosis, you can say, the parental cell has a diploid number of chromosomes. The diploid number is 2n. Meiosis, you will see, requires two cell divisions. It requires two cell divisions, meiosis 1 and meiosis 2. And this process will yield 4 daughter cells that have a haploid number of chromosomes. DNA replication is going to occur prior to meiosis 1, just like we saw with mitosis. Let's move through and let's talk meiosis 1. When we talk meiosis 1, here the chromosomes come together and they line up side by side. They line up side by side. They're going to line up side by side at the equator, at the synapsis. Now, when the homologous pairs separate, each daughter cell receives one member of the pair. The cells are now haploid cells then. Now, when we talk meiosis II and fertilization, in meiosis II, the sister chromatids separate. In meiosis II, the sister chromatids separate. Now, in the end of each of the four, each of the 4 daughter cells has the haploid number of chromosomes. In humans, these daughter cells mature into gametes, sex cells, or sperm and eggs, all the same thing. Fertilization then? Fertilization restores the diploid number of chromosomes in a cell that will develop then into a new individual. So when we look at meiosis in a little bit more detail, okay, we're going to see no replication of DNA occurs between meiosis 1 and meiosis 2. DNA replication occurred one time, and that's the only time that it happens, before meiosis 1. The centromeres divide, and sister chromatids are going to migrate to opposite poles to become individual chromosomes, we said. And then each of the 4 daughter cells produced has, we said, the haploid number of chromosomes. Each chromosome is composed of one chromatid. So here we can see we've got our 1, 2n, 4, basically diploid number cell. This cell then undergoes DNA replication, right? It goes from G1. It goes from then G1 to then the S stage. In the S stage, that's where DNA replication occurs. So look at here. We've got 4 homologous chromosomes. So here you can see we have homologous chromosome pairs. And once they undergo DNA replication, you can see here they've all doubled in quantity. So here's all the sister chromatids found in this cell that's already undergone DNA replication. Now this one cell then undergoes meiosis, giving rise to 2 separate cells. Now that's meiosis 1. Right? If this was mitosis, this is where it stopped. But here we're going to have, we said, meiosis 2 occur. Now when we talk meiosis 2, you can see then allows those 2 daughter cells to divide into then 4 daughter cells. And each cell, now look at here, has a haploid number of chromosomes. Look at here, this parent cell had 4 chromosomes. Here we can see 2 in each one. So when this one sperm cell is going to fertilize an egg cell, that will help restore then the diploid number of chromosomes. Now when we look at meiosis in greater detail, we'll be able to see everything happens step by step. But when we talk fertilization, again, fertilization, the daughter cells of meiosis mature into gametes. Daughter cells of meiosis are going to mature into gametes. So when we talk fertilization, right, we said we have the daughter cells. These daughter cells, they're going to mature into gametes. Then those gametes are going to, if we're talking sperm, they'll come together with eggs. Right, then these gametes will come together, eventually restoring the diploid number, which is what we call fertilization. So fertilization restores the diploid number, where you get one haploid cell and another haploid cell, and you put them together, giving rise to a diploid cell. So this will create a cell that will develop into a new individual. Now, when we look at meiosis in detail, I want you to know again, meiosis is going to require 2 nuclear divisions. It requires 2 nuclear divisions and results in 4 daughter nuclei, each having a haploid number of chromosomes. When we talk about the first division, meiosis 1 has 4 phases. Prophase 1, where the chromosomes come together and they're going to line side by side. We saw before in prophase 1 of mitosis, basically the same events occurring. The spindles here are going to appear. The nuclear envelope is going to fragment, and the nucleolus disappears. Well, then here we can see we have a way to induce genetic variation. Genetic variation is going to be induced a couple of different ways. Number 1, through crossing over, and second, through independent assortment. So here we can see, still in prophase 1, the genetic information is exchanged in a process called crossing over, where we see the sister chromatids then are going to no longer be identical. They will then no longer be identical. Second, then we are going to have metaphase. Metaphase, these are a little out of order. Second, then we will have metaphase. Now, in metaphase 1, we're going to see the chromosomes are going to align at the metaphase plate. They will align at the metaphase plate, just like we saw before, right at the center. Next then is anaphase. Anaphase 1 is where we see independent assortment take place. It occurs, and this is going to help generate cells with different combinations of maternal and paternal chromosomes. Independent assortment occurs, and it's a process which generates cells with different combinations of maternal and paternal chromosomes. Last then we have telophase. Telophase is when you see the nuclear envelope reforms, the nucleoli is going to reappear, The nucleoli will reappear, which then is going to be followed by cytokinesis. Here we'll have what we call interkinesis. So here we will have what we call interkinesis. Interkinesis is going to be a period between meiosis I and meiosis II. Now there's going to be no replication of the DNA is going to happen now. Okay? We're going to be able to move then. So as we move through then, here we could go through and look at all of these different stages in detail. So prophase 1, I want you to know about it in full-on detail as we went through. Here you can see the sister chromatids. And then here you can see crossing over take place. Here you can see now this chromosome you have here is going to cross over with this chromosome here. So what that's going to do is it's going to help to create 4 completely different chromosomes. So you can see here now we have one sister chromatid, another one that's fully red. This one is mostly red and a little blue. So they're not identical. And then here we can see we have 3/4 blue and 1/4 red. That is not anything like any of these ones here, nor does it look anything like this one here. So again, this is why we don't look like our siblings. We have a lot of features that may look similar, but we're not identical. So here then you can see you're going to have 4 different sister chromatids. So after prophase then we will have metaphase 1. In metaphase 1 is where the chromosomes are going to align at the metaphase plate in the center. Third, then we have anaphase 1. Anaphase 1 is where independent assortment occurs, which generates cells with different combinations of maternal and paternal chromosomes. Last then is going to be telophase 1. Telophase 1 is where the nuclear envelope reforms. The nuclear envelope reforms. The nucleoli will reappear. And this will be then followed by interkinesis. Interkinesis is going to be a period between meiosis I and meiosis II. There is no DNA replication that occurs now here again or anything. We go then to the second division. So before we jump into the second division, let's look at all of these different events then in detail. So first we'll jump into prophase and we'll look at crossing over. So here you could go through prophase in detail as we've kind of talked about. Here then we can see a nice example of crossing over where we have our sister chromatids. These sister chromatids then undergo crossing over, where then now we're going to form 4 completely different chromosomes. So you can see 1, 2, 3, and 4, and they're totally different. This one does nothing look, this one here looks nothing like this one, nor does this one look anything like this one. So here you can see 1, 2, 3, and 4. So here then we can see metaphase we went through. You can appreciate metaphase. And then taking us into anaphase. So when we talk metaphase and anaphase, here's where we move from the chromosomes being at the metaphase plate, and then moving to basically separate sides where independent assortment is taking place in anaphase. So here you can see an independent assortment. This helps to generate cells with different combinations of maternal and paternal chromosomes. So this makes our genetic variability, right, that we saw here, this genetic variation, even greater. In humans, with 23 pairs of chromosomes, the number of possible combinations you can see is huge. This value does not include genetic recombination due to crossing over. And then telophase we saw. So here you can see the cells in anaphase and then telophase. And then we've got interkinesis, the period between meiosis I and meiosis II. And we said we're not going to have any type of replication of DNA occur now. We're going to keep these cells exactly how they are, and then we're going to further divide their genetic information in meiosis II. So second division is divided into prophase 2, metaphase 2, anaphase 2, telophase 2. So when we jump into the second division, so when we move forward into the second division, here you can see the phases of meiosis 2. Prophase 2, metaphase 2, anaphase 2, telophase 2. So let's talk then prophase 2. In prophase 2, the spindles will appear and the envelope disassembles and the nucleolus is going to disappear. This leads to then metaphase 2, during which this leads us to then metaphase 2, during which the chromosomes attach to the spindle and they line up at the metaphase plate, taking us on to then anaphase 2. Anaphase 2 we see here is next. Here the chromatids separate and they move to their respective nuclei. Anaphase 2, here the chromatids will separate and move into their nuclei. Finally, then we have telophase 2. In telophase 2, we'll be able to see the spindles start to disappear. The spindles disappear and the nuclear envelope will reform. Cytokinesis then is the final step where 4 daughter cells with a haploid number of chromosomes are going to be produced. So here you can see meiosis 2, we've got prophase 2, metaphase 2, anaphase 2, telophase 2, and then we've got 4 daughter cells produced thanks to cytokinesis. When we talk about the importance of meiosis, meiosis is very important. Meiosis produces haploid cells from diploid cells. Genetic variation produces cells no longer identical to the parental cell. So again, genetic variation is going to occur a couple of different ways. First, crossing between the non-sister chromatids, which we call crossing over. And then second, the independent assortment of the chromosomes during anaphase 1. Upon fertilization, combining of chromosomes from genetically different gametes helps to ensure offsprings are not identical to the parents. This genetic variability is the main advantage of sexual reproduction. Long-term, genetic variation increases the survival of a species. So then I want you guys to go through and check out comparing meiosis with mitosis and make sure you understand fully the differences between the two. Let's jump to the human life cycle. Now, when we talk about the human life cycle requires both meiosis and mitosis. It requires both meiosis and mitosis. Now, in this human life cycle, a haploid sperm and a haploid egg join at fertilization. And then the resulting zygote has the full, or we say the diploid number of chromosomes. During development of the fetus, before birth, mitosis keeps the chromosome number constant in all the cells of the body. After birth, again, mitosis is going to be involved. Mitosis is going to be involved in the continued growth and repair. Next, then, let's look at meiosis in the testes of males. This is the process we call spermatogenesis, and it's going to be basically the process where we're going to see the production of sperm occur. And the meiosis in the ovaries of females is going to be called oogenesis, and it'll be responsible for producing eggs. So here you can see we've got a zygote, The zygote undergoes mitosis, gives rise to this human. The human then continues to have mitosis take place, and they grow up, and then you can see they have meiosis taking place in their gonads. So here meiosis occurs, helping to produce the sperm and the egg, which then will come together during fertilization to then produce another zygote, another human, and this cycle will continue. So you can see mitosis and meiosis are both equally important. So let's look at spermatogenesis. In human males, again, meiosis is a part of spermatogenesis, which occurs in the testes and produces sperm. So here, following puberty, when the sex organs mature, spermatogenesis is continual in the testes of human males. During spermatogenesis, primary spermatocytes, which are diploid cells, they will divide during the first meiotic division to form two secondary spermatocytes. Forms two spermatocytes, which are haploid. Then during the second meiotic division, four spermatid are formed. Spermatid then mature to sperm, which are haploid cells. As many as 300,000 sperm are produced per minute. So here we can see the primary spermatocyte. It undergoes meiosis 1, giving rise to two daughter cells. The secondary spermatocyte then undergo meiosis 2, giving rise to four spermatid. These spermatid undergo metamorphosis, and then they mature into four sperm. Again, haploid number of chromosomes. Next, then, let's talk about oogenesis. Meiosis here begins in the fetus. In the ovaries of the fetus, all of the primary oocytes, the diploid cells, they begin meiosis, but they become arrested, or you can say they stop in prophase 1. They become arrested in prophase 1. Following puberty and the initiation of the menstrual cycle, one primary oocyte begins to complete meiosis. It finishes meiosis. It finishes the first meiotic division as two cells, each a haploid cell. Now, one of these cells is going to be called the secondary oocyte, and it receives almost all of the cytoplasm. The other cell you see here, the other cell is called the first polar body. It's basically a non-functioning cell which eventually will disintegrate. But before it disintegrates, you'll see if it divides, it gives rise to basically other non-functional cells. So we'll see that here. We can see when it divides, it forms a second polar body. Again, a non-functional cell also will disintegrate. So here, if the secondary oocyte is fertilized by a sperm, so if the secondary oocyte is fertilized by a sperm, it completes the second meiotic division. And if it completes the second meiotic division, here, let's say this first polar body completes the second meiotic division, as does this cell. Again, you're going to have basically three polar bodies that are produced and one functioning cell here. The chromosomes of the egg and the sperm are going to unite. The chromosomes found in the nuclei of the sperm and the egg, they'll unite to form a 2N zygote. So you can see here in the male, 4 functional sperm are produced, whereas here in oogenesis, only 1 functional egg is produced. Now, if this secondary oocyte is not fertilized, if it does not get fertilized, it's going to pass out of the body along with menstrual flow. So here you can see spermatogenesis producing 4 functional cells. And then oogenesis, you can see you've got really one functional cell.